Here, we're talking about how cancer, in its essence, is it always represents a genetic disorder because it always begins with some sort of mutation in one of these social control genes, one of these genes that regulates cell division, cell differentiation, okay, DNA repair, all of these genes that contribute to the control over proliferation and differentiation. Okay. And we talked about these basic properties of cancer cells. They grow and proliferate despite the fact that there's no signal in the environment telling them to grow and proliferate. Okay. They escape signals to die. Sometimes they turn off signals for apoptosis, or they turn off signals that limit the number of times they can divide. They often resist differentiation. Okay. They tend to try to stay as undifferentiated as possible. Okay. And what is the advantage of being, from a cancer cell perspective, what is the advantage of staying undifferentiated? The mitotic activity, the rate of cell division is much higher for undifferentiated cells. Okay? So it's much easier to proliferate if you remain undifferentiated. Okay? But that also means that undifferentiated cells also don't contribute to the function of tissues. Okay? So as we'll see later when we talk about staging and grading of cancers, the cancers that tend to be the least differentiated tend to also be the most aggressive cancers. They spread the most rapidly, and they tend to be the most invasive. Okay? And they also tend to make people sicker because they contribute the least to the tissue. Okay? And the disease progresses much more rapidly. And cancers, as we talked about, are genetically unstable. They actually evolve. They evolve and accumulate mutations, okay, because they are genetically unstable. They often silence things that remove mutations or catch mutations. Okay? So they tend to actually accumulate those mutations. And cancer cells are invasive. They grow, they spread, and they take over. They invade, kill, and take over neighboring healthy tissue. Okay? And sometimes that includes not neighboring tissue, tissue that's in distant sites. Okay? So these two terms, autonomy and anaplasia, autonomy is the idea that the cancer cells need no outside cues in order to proliferate. Okay, so they need no outside cues in order to proliferate and grow, which is very different from the original host cells. And anaplasia, that term refers to a lack of differentiation, where the cancer cells in that tissue bed will look less differentiated often than the surrounding tissue. So let's talk a little bit more about this idea of cell mortality. So cell mortality really refers to the idea that any particular cell, any individual cell, it can only divide itself so many times. It can only make so many identical copies of itself. Okay? And usually that number is about 50 or 60. And once the cell makes about 50 or 60 copies, that particular unique cell stops making copies of itself. Okay? And so that is called senescence, that limit in the number of times a cell can make an identical copy So in terms of mortality, it's really about a mortality of that particular cell's set of genes, okay? Or that particular cell's versions of those genes. Okay, so what normally happens is that cells will divide and double until about 50 or 60 times, and then they stop dividing. Okay? And that's what we call senescence. Sometimes, and this is still in the realm of normal, sometimes some cells continue to divide beyond 50 or 60 times. Okay? So if you think about it, you start off with a single cell. It doubles, the daughter cells double. Okay? All of those daughter cells double. Okay? So now you have this huge population of cells, and it represents 50 or 60 cell divisions. Sometimes the cells will continue to divide beyond 50 or 60. But then what's normal is that most of those, or all of those cells beyond 50 or 60 divisions will hit what we call crisis, where all of those all of those cells will die off en masse. Okay. So all the cells that extended beyond 50 or 60 divisions should at some point hit crisis where they all die. Okay. If a cell line divides beyond 50 or 60 and doesn't hit crisis and continues to divide, those are the cells that we talk about that have become immortalized. Okay. Because the normal control that prevents proliferation beyond 50 or 60 divisions has been silenced somehow. Okay? So how does this work? Well, that's a big question, how this works. And one important piece of that answer is actually a relatively new discovery, relatively maybe in the last 10 or 20 years. Okay? There's a brilliant woman by the name of Elizabeth Blackburn who discovered an enzyme called telomerase in the cells of at least humans, probably beyond humans. Okay? Now, telomerase, it's an enzyme whose function is to rebuild a portion of DNA strands called telomeres. Okay? So at the ends of DNA strands are these segments called telomeres. And the telomeres are very much like those little plastic tips on shoelaces. Okay? What do those little plastic tips on shoelaces prevent the shoelaces from doing? From fraying, right? So when those little plastic tips fall apart, all of a sudden the ends of your shoelaces look like that. Right? So the telomeres prevent the DNA strands at the very ends from separating. Okay? Because when they start to separate, then those portions of DNA become vulnerable to mutation. Okay? So these telomeres, every time the DNA replicates, right? Every time that cell divides and the DNA replicates, those telomeres get shorter. Okay? So every time the cell replicates and the DNA doubles or replicates, those telomeres get shorter. And the function of the enzyme telomerase is to rebuild those telomeres. But a cell's ability to to produce telomerase progressively declines with the number of cell divisions it undergoes. Okay? So with each cell generation, that cell's ability to produce telomerase and repair their telomeres gets lower and lower until they finally run out of telomerase and can't rebuild their telomeres. Okay? And when they stop rebuilding their telomeres and the telomeres get short enough, errors start to accumulate along the terminal portions of that DNA. Okay? And as we talked about last time, those DNA repair proteins bind to those errors and block replication. They block cell division. These DNA repair proteins, they bind to those errors and they block cell division and replication. So that's talking about that. That's senescence.
The telomerase enzyme helps rebuild the telomerase.
Yes, yeah. So when the cell runs out of that telomerase, the telomeres shorten. Okay? Yes, sir?
As they keep shortening, is the accumulation of errors what signals the cell line to die?
So the accumulation of errors signals the halting of the division. Yeah, but that signals the senescence. Okay. So for the cells that escape that senescence, somehow they just continue to multiply. All of those daughter cells now have lots of errors, right? And if you recall, I talked about at least one protein that starts to get expressed when the cell finds errors, P53, right? And when there's enough of that P53, what happens to the cells? It should happen to the cell. Apoptosis. So that's crisis. So those cells that escape senescence, they have lots of errors. They start accumulating that P53, and when they have enough of that expression, they trigger apoptosis, and all those daughter cells die. Okay. So one mechanism of getting immortalized is a cell's ability to overexpress telomerase. So if the gene that codes for telomerase is mutated in a way that causes overexpression, then the cell, excuse me, don't do that. Go find another way to go through. Thank you. Seriously? You're going to walk through a class? They do that every single time. What's that? Not today. So, as I was saying, thank you. So if the gene that codes for telomerase acquires a mutation that overexpresses that gene, okay? Right? Stimulates that gene, overexpresses that gene. Now the cell can, in an unlimited way, repair its telomeres and continue to double as many times as it wants. Okay? So it escapes senescence, it escapes crisis. And if in addition, right, even if it hasn't developed that telomerase deal, if it continues to accumulate errors, but it's shut off p53, or it's shut off DNA repair proteins, then it won't get to that crisis, especially if it also shuts off apoptosis. So becoming immortalized, there's several possible routes to becoming immortalized, but this telomerase route is a really important mechanism, okay, for a cell line to become immortalized, these cancer cell lines. Okay. The other implication for telomerase outside of cancer is understanding cell aging. Okay. The idea that that cells age can be linked to the amount of telomerase that cell line expresses. Okay? So there are lots of stressors that actually accelerate cell aging. Okay? A lot of stressors in the form of nutritional stressors, chemical exposure, okay? And one important stressor actually is stress, right? That increases cell aging. And that means that you can have a cell line that maybe is only in its 10th generation, but it has a telomerase expression that's equivalent to being in its 30th generation. Okay? And so what that means is that tissues in your body can't repair as well. You're smiling because... Yes, sorry. No, it's not your life age. Well, actually, it does have implications on life aging, but it actually increases the risk for tissue damage, tissue dysfunction, and development of disease. Okay? And what's not great news for any of us in this room is that caregivers actually have accelerated cell aging. This is one of the studies that Elizabeth Blackburn. Yeah, I should have told you before you applied, right, to the program. But yeah, caregivers. It's especially true for people who are caring for loved ones with profound disabilities or injuries or illnesses. So spouses or parents that are caring for people. And that cell aging is actually quite dramatic. So the discovery of telomerase had a lot of implications for understanding both how cancer cells escape senescence and also how cell aging really works. Okay, questions about this before we move on to structural functional differences? Yeah. It's a reflection of its age, yeah. The amount of telomerase a cell expresses reflects sort of where it is generationally in that aging process, yeah.
I got a little lost when you were saying, like, can you just clarify how telomerase prohibits senescence? Is that what you were saying?
If a cell overexpresses telomerase, that will prevent senescence from being able to happen.
And when cells have enough telomerase, then it makes senescence.
When the cell runs out of telomerase, senescence is is not far behind.
Okay. And that's followed by crisis.
It can be followed by crisis. It doesn't have to be. Crisis doesn't happen all the time. It happens sometimes.
Okay.
Yeah.
And then what would prevent crisis from happening?
What would prohibit crisis from happening could be the telomerase. The telomerase continues to proliferate, and or that the errors that, if they have accumulated, they're not triggering apoptosis, which could be a problem with p53. It could be a problem with DNA repair. It could be a problem with apoptosis. Yes, ma'am.
So
Does the stress lead to that?
No,
Or
I
does
think that similar response where like you have an aging cell that's not actually that old because of the telomeres.
Yes, yes. Accelerated aging can express itself in a lot of ways, like triggering the onset of neurodegenerative, of disorders where the tissue actually looks much older than it should, right? Absolutely. Yes, ma'am.
Telomerase decreases as the cell ages, or is it
You said decrease? Yes. Telomerase expression, the ability to make telomerase, should decrease as the cell ages.
that's normal?
That's normal.
Patients, the opposite.
Exactly. So if part of what makes a cancer cell cancer is this mutation in the telomerase gene, right, and the nature of the mutation is an overstimulation, then what are we calling the telomerase gene? What was the term we learned last week then? A proto-oncogene. A proto-oncogene, right? Yes, a proto-oncogene is the unmutated form of a gene that, when overexpressed, produces an oncogene. Okay. What was the name of the gene that, when shut off, produces an oncogene? Tumor suppressor genes. Okay, very good. Okay. So cancer cells are obviously different in many important ways from their original host cells, right? Because all cancer cells in our body were originally normal host cells. Now, we have structural changes, we have functional changes. Some of the changes are really physically obvious when you look at the cell. So if we look for a second, over here we've got like split personality cell. Okay, we've got over here we've got the normal cell. Over here we have the malignant cell. Okay. Now, one of the things you might notice is that the normal cell has a regular shape, right? Has a nice regular shape, as opposed to this cancer cell, malignant cell, that is very amorphous. Okay, it's very globular in some places. Now, that regularity in shape is because of a disorganized cytoskeleton. Okay, remember that scaffolding that is the interior support system for cells. It gets disassembled when the cell mitotically divides. Right? The cytoskeleton becomes part of that mitotic spindle that helps line up the chromosomes and then pull the chromosomes apart. Okay? And so that means that the cytoskeleton is very disorganized. Okay? So this malignant cell having a high mitotic rate is going to have a disorganized cytoskeleton in an irregular shape. Okay? The same is true with the look of its nucleus. Okay? The nucleus is also going to look very disorganized, okay? Going to have an amorphous shape for the same reason. Okay? The chromosomes and the chromatin are unwinding. They're in some stage of DNA replication and cell division, and they just look very disorganized. Okay? So over here, we've got cell adhesion proteins or cell adhesion molecules. And over here, what they're showing on the normal cell is that they have these cell adhesion molecules that anchor one cell to the neighboring cell in the tissue. Okay? One important example of these anchoring proteins is called fibronectin. Okay? Fibronectin. Now, these proteins do exactly what is illustrated. They anchor the cells to each other, anchors the cell to the extracellular matrix, okay? And it helps maintain the integrity of the tissue. But it also does something else. By being physically connected to your neighbors, you're also aware of how many neighbors you have. when a cell in a tissue bed is physically attached to its neighbors, it also knows how many neighbors it has, which is going to tell the cell something about the cell density of the tissue. Okay. And when cells reach an optimal density in that tissue, replication and division, bless you, are strongly inhibited. Okay. So when a tissue bed reaches a particular optimal cell density, the replication or division shuts off. This used to be called contact inhibition, and now we call it density-dependent growth. It's called density-dependent growth. You don't need to learn the old irrelevant term for it. But this is this notion, if you recall this at all in some of your previous biology classes, this is the idea that if you are growing a cell in culture, let's say, a little petri dish, you're growing a cell in a tissue culture lab, a normal non-malignant cell population will grow until it grows into this nice, neat lawn, right, of cells on the petri dish. Cancer cells, on the other hand, don't stop at the nice, neat lawn. They just continue to grow. They grow on top of each other, and they will grow until all of the food or the media underneath it is completely gone. Okay? They'll use up all the resources, and then they'll all die. Okay? So for cancer cells, they often lack these adhesion molecules. They often lack these adhesion molecules, including fibronectin. So they don't care what the density of the tissue is. That does not control their growth. And it also means that cancer cells in a tumor are not anchored to one another. So it makes them much more mobile. Okay. They will often just simply fall off or shed off of a tumor. Okay. If you think about ovarian cancer is most commonly from the external epithelial lining of the ovaries. Did I say ovarian? Yeah, ovarian. So the external epithelial lining of the ovaries are usually the original cell that develops the malignancy with ovarian cancer. So ovarian cancer actually has a very unique method of spread because the tumors are actually on the outside of the ovaries, and the way that they spread is they just fall off into the pelvic cavity and fall off into the into the abdominal cavity, and then wherever they fall, they can form a metastasis. Okay. And it's a very insidious form of cancer because some of the only symptoms that people will have is abdominal bloating, like a sense of abdominal fullness, right? And unfortunately, the vast majority of ovarian cancers, by the time they're caught, are very, very late stage. They're usually at least stage 2, if not 3 and 4. Okay. So these cells of the tumor, because they're not attached to one another, they are very mobile, and that aids their ability to spread and invade. And then in addition to not being attached to each other and therefore very mobile, they also have the ability to release lysosomal enzymes and degrade surrounding tissue and literally also produce and release lysosomal enzymes, those digestive enzymes, cancer cells. And that allows them to eat away, degrade the surrounding tissue. Okay. It also allows them to invade lymphatic vessels and get into the lymph flow, invade blood vessels and get into the bloodstream. Okay. And once they're there, they can travel and get to a distant site and eat through the blood vessel there and get into the tissue. Okay. So cancer cells are mobile, and they produce these and release these lysosomal enzymes that help them invade and spread. Yeah.
So all of these changes are coming about from some mutation that going back to it, like all of these things come to that.
Yeah. So the cytoskeletal changes and the nuclear changes are mostly about being mitotically active, but the lack of cell adhesion and the ability to increase your production of lysosomal enzymes are common mutations of malignant cells that allow them to spread. They're not going to be found in all malignant cells, but many of them. Yeah. Okay. So what are some of these mechanisms for mutagenesis, meaning the genesis or the development of mutations, okay, that can cause malignancy? By far the most common form of route to mutagenesis is this very nebulous thing that we call gene-environment interactions. And this is the idea that there's something in the environment, okay, something in the environment that is altering genes, leading to mutations, and leading to malignancies. Okay. And certainly some things in the environment, excuse me, some things in the environment we know, we've identified over time, are what we call carcinogenic, which means that they cause mutations that tend to produce carcinomas or malignant cells. Okay? So lots of things we've identified as carcinogenic. About half of the ingredients that's added to tobacco in a normal cigarette, carcinogenic. Right? We know that. A lot of the, so ionizing radiation, carcinogenic. Okay? So a lot of the things that we are exposed to, we know are carcinogenic. We try to minimize the exposure. Okay? UV light from exposure to the sun increases the risk of all types of skin cancer. Okay? So there are lots of things in the environment that we know have mutagenic properties. And then there are some things that are connected to certain environments that seem to produce higher rates of certain cancers, but that we don't really know what those things are. Okay? So, for example, if you look at the same population, same patient population, so women in their reproductive years, okay, if you take a look at women in Japan versus women in the United States, women in Japan of the same age, of the same everything else, have a much higher risk for stomach cancer than in the United States. Okay? And conversely, women in the United States have a much higher risk for breast cancer than the same age woman in Japan. Now, if you sort of swap women, so if someone in the United States moves to Japan, someone in Japan moves to the United States, and they both stay in these new settings for a long period of time, their risks swap. Now, the American woman in Japan has the same stomach cancer risk as the rest of the Japanese women and the lower breast cancer risk. And the Japanese woman in the United States now has a higher breast cancer risk and lower stomach cancer risk. Okay? So it's a cancer-specific risk that is very much tied to some location, right? Some particular environment, but we don't know what in the environment is causing it. Okay? So gene-environment interactions by far is the most common cause of cancers or mutagenic mechanism. Inherited cancers, okay, inherited cancers are actually not that common in terms of all causes of cancer. Okay? However, it's a cause of cancer that's under a lot of investigation because if we can identify the genes, we can screen people. Okay? So it's a target for a lot of intense research and trying to understand it because of the potential screening tool, okay, for the ability to screen people and prevent the cancers. So BRCA1, BRCA2, like I talked about last time, is an excellent example of an inherited cancer gene that you can scan for, or you can screen for, and certainly will increase, especially among women, increase lifelong risk of certain cancers, especially breast cancer, okay, and ovarian cancer as well. So it's gotten to the point where women especially who are identified with BRCA1 mutations, preventive double mastectomies is actually very, very common, right? You read probably in the New York Times, what was it, a year or two ago, Angelina Jolie had both breasts removed and reconstructive surgery prophylactically because they found out she had a BRCA1 mutation. Okay? Also, what sometimes is done is, in addition to the mastectomies, a total hysterectomy is often done as well, which is, of course, by the choice of the patient, but it's it's a choice that is driven by sort of peace of mind. Right? So inherited cancer genes, like I said, are not as common compared to other causes of cancer, but they are an important one to understand for preventive reasons and screening reasons. And as I mentioned before, nearly every inherited cancer stems from an inherited tumor suppressor gene or mutated tumor suppressor gene. Okay. That's the most common form of these inherited cancer genes. Yes, ma'am?
I assume that with most pediatric cancers,
For most pediatric cancers, are they forms of inherited cancers? I don't know. And I wouldn't necessarily assume that. I wouldn't necessarily assume that. Certainly going to make up some. I don't know how much. Yeah. Okay. Oncogenic viruses. Okay. Oncogenic viruses. Now, the most common mechanism for mutagenesis for an oncogenic virus is something called direct or insertional mutagenesis. Okay? And that's when a virus infects a particular tissue bed. And of course, viruses are going to get inside of cells, and they're going to use cells to replicate the virus. So oncogenic viruses are viruses that in the process of infecting that host cell, they insert an oncogene into the cell's genome that gets propagated. Okay? So they insert an oncogene into the host cell's genome that gets propagated. And they transform the cell. Okay? They cause the cell to become cancerous by adding that oncogene. So this is an example of when an oncogene is not a mutated form of a normal gene. Because the oncogene is coming from the virus. So an excellent example, or very important example of this is the human papillomavirus. Right? So human papillomavirus, a sexually transmitted disease, often infects cervical cells and is implicated in causing potentially up to 70% of the types of cervical cancers that develop. Okay. And so there's a big push now for getting young girls and boys vaccinated for HPV in order to reduce the likelihood of spread of the virus and preventing cervical cancer as women get older. Okay. So that's the direct mutagenesis or insertional mutagenesis. Okay. There's also an indirect way that viruses can lead to mutagenesis. And in this way, the virus infects this person's body, attacks a tissue bed, and essentially it contributes to a lack of immune regulation that would normally catch the mutation. So the viral infection is preventing the immune system from being able to recognize the fact that a cell has become transformed and attacking the cell. Okay. One very, very clear example of this is the development of Kaposi sarcoma in patients with HIV. And actually, if you've got Kaposi sarcoma, you've got full-blown AIDS at that point. Right? Kaposi sarcoma, KP, sometimes called the Kaposi sarcoma. But it's a very, very rare form of skin cancer that is caused when someone's immune system is very, very weak and can no longer kind of regulate cell proliferation in the skin. Okay. But there are also oncogenic bacteria, so bacteria that tend to infect a tissue bed for a prolonged period of time, and as a result increase the risk of developing cancer. Okay. So an example of this is H. pylori. Okay. So H. pylori, which is a funny bug. H. pylori is a bacteria that's found in about half of people, the digestive systems of about half of people in the developed world. Okay? And only a small percentage of those people, H. pylori is highly, having H. pylori colonization increases your risk for development of peptic ulcers. But only about 10-15% of people with H. pylori ever develop peptic ulcers. But H. pylori has the ability to live in the stomach, live in the duodenum, okay, live in your GI tract, and because it does not really cause a lot of cell injury, it doesn't really trigger a huge immune response. But it certainly will trigger some inflammation. Okay. And whenever whenever you have inflammation that's prolonged over a period of time, it really represents a chronic injury. And tissue beds that undergo chronic injury are vulnerable to development of cancer. And that's an important thing to understand, is that any tissue bed that has sort of a chronic infection, a chronic injury, has nearly any tissue bed has the increased vulnerability of developing cancer, okay? Especially if there is epithelial tissue in that region, like there is in the GI tract. And the reason is that with chronic injury and chronic inflammation, there's also chronic cell turnover. You get injured tissue, the cells try to regenerate, they try to heal that tissue. Okay. And whenever you have lots of cell turnover, you run the risk of mutations. Okay. So people with chronic H. pylori infections, especially the ones that tend to cause peptic ulcers, they have an increased risk for carcinomas or cancers of the stomach, especially of the stomach. People with chronic hepatitis, that especially if it leads to cirrhosis, an increased risk of liver cancer. So where any of these tissue beds where there is a high regenerative ability, it increases the risk for development of cancer. Okay. And sometimes that chronic injury is from the presence of a chronic viral infection or chronic bacterial infection. Does that make sense? Yes, ma'am. Yes. Yes. So the indirect mutagenesis through the viral infection and oncogenic bacteria, they're working through very similar mechanisms. Yes. Okay. All right, so clinically, when cancers are identified, they are given a designation. They're given two basic designations. They are graded and they are staged. So whenever a cancer is identified, the cancer is graded and it's given a stage. The grading of a cancer is done through the examination of the tumor cells. Okay, so normally a biopsy is done. The cells are visually examined, and they are given a grade that reflects their degree of malignancy, okay, or how aggressive this cancer is. Okay. High grade refers to highly aggressive, low grade, less aggressive. Okay. So aggressiveness is really about the potential for spread. Okay. So if a cell has a high turnover rate, if a tumor has a high turnover rate, it's very mitotically active, and it accumulates new cells at a high rate. If it's very undifferentiated, those tend to cause it to be a high grade. Okay. If cells have a lower, relatively low mitotic rate, they're not accumulating cells very quickly, bless you, it's fairly well differentiated, then that could get it a designation of a low grade. Okay. Yes?
So, how does it progress? You get, if you're right
often at first diagnosis, there'll be a grading of the cancer, and then so sometimes this happens often with prostate cancer, where some prostate cancers are, when they're initially looked at, they're assessed to be very low grade. Okay? And so some prostate cancers actually grow extremely slowly, and they are of very little clinical importance. Okay? So what they do is they do oftentimes this thing called active surveillance, where they will periodically do PSA testing, which tests the cellularity of cancer cells in prostate cancer cells in the body. They will actively monitor any change in prostate cancer symptoms, right? And if there is a change, they might do another biopsy to assess if the grade has changed. Right? Or if a primary tumor, if you develop a new metastasis in an area, often that secondary tumor will be biopsied to see if the grade has changed. Okay? But the grading of a cancer is an important initial step to potentially predicting the rate at which it's going to spread, and therefore how aggressive the treatment needs to be. Yes?
How do you label a grade?
It's often high grade, low grade.
So it's just higher.
Usually high or low. Yeah. And there might be, for different cancers, there might be a numerical system attached to that, but often you'll hear it referred to as high grade, low grade.
Okay.
Yeah.
And the stages, when it's like 1, 2.
Right. The stages always have a numerical staging system. Okay. So to stage a cancer, really what we're talking about is what stage in the spread of this cancer is the patient in. Okay. And different cancers have different staging schemes. Okay. Because different cancers spread through different patterns. Okay. So, for example, when I talked about ovarian cancer, if ovarian cancer is which it's rarely caught at stage 1, but stage 1 represents no spread outside of the ovaries. Okay? As soon as it's spread to another organ, it's stage 2, and if it's spread to more than one organ, it's stage 3. And if it's spread to, I believe if it's spread to like the liver or the diaphragm, it's at stage 4. Okay? So it all depends on different, and I may have gotten some of that wrong, but you get what I'm saying. So different cancer types have different patterns of spread and will be staged accordingly. Okay? And generally speaking, the higher the staging number, the farther the spread has gotten, the much more difficult the treatment is, and potentially the poorer the prognosis. Stage 4 cancers across the board tend to be thought of as terminal. Okay? And so usually at that point, the treatments are palliative, mostly. Okay? And palliative, if you haven't come across that term yet, palliative treatment is not a curative treatment. It's a treatment that's meant for potentially extending life or enhancing comfort at the end of your life. Okay. Yes, ma'am. When are they feeling, for ovarian cancer, when are they feeling the abdominal fullness? Often when it's at stage 2 or 3, which is why it's some of the first, it's usually when it's first discovered. You know, if someone has a BRCA mutation, they may actually be incredibly sensitive to any kind of abdominal bloating, and it might be caught much earlier. Or there might be sort of active surveillance of that if they're in a high risk group. But usually, unfortunately, it's a very difficult cancer to catch and cure because of that. Although I've I've known people who were caught at stage 3, total hysterectomy along with aggressive chemo. This person's doing great, right? So it's not a guarantee, but it's much harder once it's spread to that far. Yes.
Like things like blood cancers, how would you grade stage blood cancer?
That's a really good question. So leukemias, blood cancers, the staging is much different, and it's usually symptomatic because the cancer cells are replacing often bone marrow cells and blood-borne cells. So the staging is often going to be about the degree of symptom manifestation as a result of non-functional cells. Yeah, that's a really good question. But the staging is really about the degree of spread, and it's very cancer type specific, okay, depending on the type of cancer you're talking about. Okay, so when we talk about going from the transformation of a cell to the formation of a tumor to the spread of that tumor, there are a couple of characteristics that are unique to cancer cells. Okay? So when we talk about tumors, there's a very big difference, both obviously in terms of implications, but also in terms of physical appearance and behavior of malignant tumors compared to benign tumors. Okay? A benign tumor is a tumor that really it's inappropriate. It means it shouldn't be there. However, it's non-invasive. Okay? It doesn't have potential for rapid spread. It's really not malignant. It's really not aggressive, this tumor. Okay? So a benign tumor is often an overgrowth of the original tissue type. Okay? So a benign tumor is often an overgrowth of the original tissue type. So uterine fibroids are a really good example of just such a run-of-the-mill benign tumor. Okay? Uterine fibroids are usually composed of the same muscle type as the uterine muscle itself. Okay? So it's an overgrowth, it's isolated overgrowth of often uterine muscle. Okay? Some of the features are that the appearance of the benign tumor is very regular, and it conforms identically to the original tissue type. Okay? They look like very well-behaved cells. They just shouldn't really be there. The other thing about benign tumors is they're often fully encapsulated. Okay? And they don't destroy surrounding tissue, they just displace surrounding tissue. Okay? So if you can see this benign tumor, this is actually most likely a lipoma. Does anybody have these on their body, like just at the skin? Yeah, they're weird, right? My husband's got a ton of lipomas. I just constantly play with them. They're really, I just can't help it. I just constantly play with them. And some of them are painful, so he doesn't really appreciate it. But, so this is like a classic lipoma, where you've got just, for some reason, this encapsulated amount of fat in the skin. It will often kind of sometimes have a raised appearance because it's displacing the skin tissue. It's not eating a hole around it. And it's not, except if it's painful and you want it removed, there's no reason to get a lipoma removed because it doesn't have any potential for spread or becoming cancerous in any way. Okay. Yes? Benign, you never say never in biology for sure, right, and in medicine. But benign tumors, like fibroid tumors in the uterus, they have like a 0.5% chance, right, of turning into cancerous tissue. Okay? They are abnormal, and they represent abnormal proliferation of cells, which is where the potential stops, right? But they have none of the features that we've been talking about. Yeah. Okay, I'll come back to some more questions. But so a malignant tumor, on the other hand, okay, you notice there's nothing regular about the malignant tumor's borders. Okay, very irregular borders. For this particular malignant tumor, it has eaten its way to the surface of the skin, and so it has ulcerated the skin. And so this ulceration will often manifest as like a sore that doesn't seem to want to heal. Right? No matter how long you've had it, it doesn't seem to be healing. You notice that this malignant tumor does not displace the surrounding tissue. It has eaten its way through the surrounding tissue. You also notice that there is a area of necrosis or dead tissue, which is very common for tumors. Okay? Just because the cells are multiplying rapidly doesn't mean that some of the tumor cells aren't also dying. Okay? In fact, oftentimes the accumulation of mutations increases the likelihood that the tumor cells are going to eventually die in the tumor. So having areas of necrosis are not uncommon. Okay? One thing that you can't see with this tumor, it's not drawn here, but these tumors will often have blood vessels that have sprouted to perfuse them directly. So they'll be highly vascularized, the tumors. And if you notice that this tumor is extending out and it's invading a local lymphatic vessel, it's invading a local blood vessel. So oftentimes, this spread to the lymphatic system and to the circulatory system is of great concern, because then cells can break off the tumor and travel to a distant site and form a secondary metastasis, which represents a stage of spread. Okay? And oftentimes when a tumor is identified in the body, if the tumor is being removed or being biopsied, what will often be biopsied as well is something called a sentinel lymph node. And the sentinel lymph node is the nearest major lymph node to the site of the tumor. Because if cancer cells have managed to get into the lymphatic system, the first place you'll see those cancer cells oftentimes will be that nearest lymph node. Okay? So oftentimes when breast cancer is found and either the lump is removed or the breast is removed, the sentinel node will be biopsied, and if it's found to be, if cancer cells are found there, in addition to the removal of the breast or the lump, chemotherapy or radiation will also be recommended. Okay? Even if a secondary tumor hasn't been clearly identified. Okay? So I mentioned about this sprouting of new blood vessels to a tumor. Now, tumors are incapable of growing more than a few millimeters in size without creating their own blood supply. Okay? So This process of creating new blood vessels is called angiogenesis. It's a process that all cells in your body, all tissue in your body has the potential for. Okay. And what stimulates angiogenesis is chronic, sort of low-level ischemia to a tissue bed. So when, for example, when people start to build muscle mass, let's say they're training, they're lifting weights, they're trying to build their muscle mass. As that muscle mass increases, okay, you're going to get to a point where the muscle tissue starts to get far away enough from the nearest blood vessels that they're not really getting enough oxygen. And they start to just become slightly ischemic, slightly hypoxic. So then they release these angiogenic factors. They're very similar to cytokines, but they release these factors that stimulate nearby blood vessels to sprout new branches towards that tissue. And so along with that elaboration of muscle tissue, you'll start to see elaboration of vasculature in that area as well. People with chronic ischemia in cardiac muscle, so people with chronic ischemic heart disease, will often, we call it, in cardiovascular physiology, they often call it the development of collateral circulation, where you'll actually form new blood vessels to perfuse the area to compensate for potentially blocked vessels that are not sending enough blood to a region of the heart. Okay, so this is a normal process that is sort of built into our bodies. But the tumor cells, they're growing out of control, right? So they quickly outgrow the blood supply that's already there. And once they reach that mild hypoxic state, they release tons of these angiogenic factors, and they stimulate the production of blood vessels directly towards the tumor. And once those blood vessels become nice and robust, the tumor growth can really continue to accelerate. Chemotherapy and radiation doesn't prevent angiogenesis. However, there are drugs that are often given along with the chemotherapeutic cocktail that are anti-angiogenic drugs. And they're not going to kill the tumor. They're just going to slow down or stop the growth of the tumor, or they're going to try to do that. Yes, ma'am? No. The benign tumors are generally not becoming overly vascularized because they have normal control regulations, right? So they've overproliferated, but they get to a point where if they've outgrown the blood supply, they will often just die, right? Sometimes uterine fibroids that exist. So I had this with my first pregnancy with my daughter, that uterine fibroids, I had imperceivable fibroids. This is a lot of TMI, but I had really imperceivable fibroids before I got pregnant. But then the first trimester, your hormones are crazy, so they grew really big, right? But then when the placenta was really well developed, all of a sudden all the blood went to my daughter, and the fibroids were like, I don't like this at all, right? So that's not what they said, but but the fibroids weren't getting enough blood. And so what happened was they started to die. And that caused a lot of pain. It's called fibroid degeneration. And so it's like the fibroid tissue actually starts to die because it doesn't, there's just something about it that prevents it from crossing that line and causing the sprouting of new blood vessels. Just stretching? Yeah. Okay. So, but very good question. Okay, so last cancer slide, and we'll take a little break. So there's a lot of terms that are used when we're talking about cancer, and it's really terms that describe cancer types based on the original tissue type. Okay? So carcinoma is a really common type of cancer, and it's a really common term to describe cancers. But carcinomas are cancers that arise from any kind of epithelial tissue. Okay? And since epithelial tissue is constantly renewed, it's very mitotically active compared to other tissue types in the body, it's a little more vulnerable for developing cancers than other tissue types. Okay? So we've got stomach carcinoma, we've got lung carcinoma. You've got anywhere where you've got epithelial cells. If those epithelial cells develop into cancerous cells, you term that carcinoma. A sarcoma is a cancer of connective or supportive tissue. This is like bone, fat, muscle, blood vessels. Sarcomas are relatively rare. Okay? They're really relatively rare because those tissue types are not very proliferative in general. They're not mitotically active tissue beds. So they represent relatively uncommon cancer types. But they do exist. Lymphoma and leukemias are malignant tumors that are derived from blood or bone marrow cells. Okay? Blood or bone marrow cells. Okay? So leukemias come in a few different forms. The two major types of leukemias, lymphogenous, myelogenous, refer to the different cell lines, right? The myeloid versus lymphoid cell lines, right? And then if you've ever heard of, there's actually 4 specific types of leukemia. Acute lymphogenous, chronic lymphogenous, acute myelogenous, chronic myelogenous. Okay, those are the 4 specific types of leukemias. Okay, so obviously lymphogenous versus myelogenous refers to the cell lines. And acute versus chronic refers to how fast the disease progression occurs. And it's very much linked to how undifferentiated the carcinogenic cells are, excuse me, the malignant cells are. Okay. So acute and chronic refers to how fast the leukemia progresses. Okay. Obviously, acute leukemias progress very fast because they tend to be very non-differentiated cells of that line. Okay. Chronic versions of the leukemias are more differentiated, so the disease progression takes much longer. Okay. And lymphomas, the cancer tends to be, instead of being isolated into the bone marrow, lymphomas tend to be isolated into secondary lymphoid tissue, especially common is finding it in lymph nodes, okay, finding the cancer in lymph nodes. Mesotheliomas, okay, if you've ever spent a sick day watching bad TV, you've seen lots of ads for shyster lawyers for mesothelioma claims. But these are tumors that are derived from basically serous membranes. So mesothelial cells that line the peritoneal cavities, the peritoneal membranes and the pleural membranes. Okay. So it's a very special particular cell tumor type. Gliomas and astrocytomas are tumors that are derived from brain cells, specifically from glial cells in the brain. Okay. And all, as I said last time, all primary brain tumors are glial in origin. Okay. You don't see primary brain tumors being of neuronal origin. Germ cell tumors are tumors that are derived from germ cells, normally found in the testes or the ovaries. Okay. But as I mentioned, like ovarian cancer, they're not germ cell tumors usually. Ovarian cancers are actually carcinomas because they are derived from the external epithelium of the ovary, most commonly. Okay. This means that they are the cells that turn into sperm, turn into ova. That's what a germ cell tumor would be. It would just be a particular type of ovarian or testicular cancer. Yeah. But very rare, not very common. Melanoma is a malignant tumor originating as melanocytes. So the pigment-forming cells in the skin, in the dermis actually, can sometimes form malignant tumors that we call melanomas. So melanoma is a type of skin cancer that originates as the melanocytes, which is not the most common type of skin cancer. The most common type of skin cancer is something called squamous cell carcinoma, right? Which actually arise from the external epithelium, right? The squamous cells that form your epidermis. Okay. So it's most common, squamous cell carcinoma. Melanoma is a much more serious form of skin cancer, and it's originating it from the melanocytes. And then adenocarcinomas, notice that there's the root of this word is carcinoma. But an adenocarcinoma is a form of carcinoma, but it originates in glandular tissue. Okay. So these are often exocrine glands. So most colon cancers are adenocarcinomas. Okay? And they could originate from those mucus-forming cells, right, in the lining of the colon. Okay? So an adenocarcinoma, it's a type of carcinoma because the cells are glandular tissue, which is a type of epithelial tissue. Most breast cancers are adenocarcinomas because it arises from mammary